I want to talk about chapter 4 of my new book, The Quest for New Knowledge. And the title of this chapter is The Hormones of the Immune System. Because by 1980, that's where we left off in chapter 3, it was clear that we were clear to me anyway, that we were on the threshold of a whole new molecular regulatory system as to how the immune system was functioning. And that was that it was no different from any other hormonal system in the body. Hormones are molecules that are secreted by usually organs that circulate in the body and the blood and the lymphatics and so forth, and usually have target tissues or cells in other parts of the body. And hormones mediate their work by attaching to specific receptors on the surface of these cells, which then activates a biochemical mechanism that tells the cells what to do. And that's exactly what we surmised as to what the immune system was all about. And therefore, I figured that the next series of experiments over the next several years were going to be extremely important in order to establish the paradigm that the immune system is a hormonal system. And of course, the immunologists never heard of the endocrine system, as I've said before, and hormones, and they didn't really care about them and so forth. But for me, as educated as a physician, I was well schooled in what hormones were all about, and so they were, like falling off a log. And also pharmacology, the study of drugs and how they affect the body and how the body affects them is also very similar to endocrinology, because most of the drugs, most if not all of the drugs that have been created over the years, the last 200-300 years, work just like hormones do, by binding to specific receptors inside cells, inside or on the surface of cells, and then telling the cells what to do. So it's a communication system, essentially. And the 1980s were really the golden age for interleukins. I mean, this was the decade that interleukins were put on the map. Many new interleukins were discovered throughout the 1980s by many different groups. With the advantage of gene cloning, genetic engineering, you could identify and isolate the genes that encoded the interleukins, and that allowed you then to use the genetic engineering to grow up bacteria that contained your gene that you put into them, and then the bacteria would make huge quantities of the protein, and you could and since bacterial culture is very simple by comparison to cell cultures of nucleated cells, you grow up vats of these bugs, of these bacteria, and they will then spew out the protein, and then you can then take the culture fluid that these bugs are growing in, and you can purify the protein, and you can give milligrams and kilograms of your favorite molecule. And that was never possible before this. But before this, if you go back in the history of biochemistry, the poor biochemists spent all their time in the cold room, because they'd go to the slaughterhouse and they'd get beef hearts, like, kilograms of beef hearts. And they would take them into the cold room, and because they didn't want the proteins to break down and to denature, and then stick them through wearing blenders and grind them all up and so forth and so on. And then then they'd end up with this big slurry, beef heart slurry, that they would then put through several different kinds of biochemical separative procedures. They start out with kilograms, and they'd end up with micrograms of the stuff, hopefully pure. Not enough to really do much with. So this gene cloning thing was a revolution, and for interleukins, it was just a piece of cake after 1980, after the initial things that we and others had done in the 70s, 60s and the 70s. So what that did was it brought all kinds of new people into the business that hadn't been there before. And in particular, in the United States immunologists sort of lagged behind other people in the world, particularly the Japanese. The guys in the States, they were still chasing their holy grail of what the antigen receptor is. And they were so fixated on the T-cell antigen receptor that they didn't care about anything else. That's how we got to lymphedrec and stuff like that. So I ended up, the Japanese, when they got in the interleukin story in a big time. And so they had at least one conference over there every year. And I went to Japan every year throughout the 1980s. And so throughout that decade, I visited every island, every major city, and made lots of friends and some frenemies as well, because we had, competitors over there. They wanted to know what I was doing, and I wanted to know what those guys were doing, So that was all very, very good. And so I was very busy in the 1980s. I was traveling a lot. I was going to meetings and seminars, giving seminars at universities and stuff. And so I was really basically, as I said in the last video on chapter 3, after the second metamorphosis, the second coming, so to speak, I was just on 24/7, and I didn't stop. I was like a little top that had been wound up and never ran out, Now, meanwhile, the family in Hanover, the kids grew up during the decade of the 80s. So, and they, went to middle school and then into high school and so forth. And they thrived. They did very, very well. And so I was busy, and the kids were doing what kids do. And we liked our new house on Pleasant Street in Hanover. So, but Lynn was, as the kids got a little bit older, and as her cooking school sort of went on, she got to an asymptote of productivity, or she basically had designed and given all the courses that she could think of to the people in the Upper Valley. And I think if she had given more, the people would have kept on taking more courses. But she wasn't really, it wasn't a profitable kind of deal at that point in time. And so she was looking for something else to do. And then lo and behold, one of her rich bachelor uncles had left her a small inheritance. So I'll never forget. I was out on the porch in one of those swings that I told you about. She came out onto the porch and she said, "I'm going to start a restaurant." So I said, okay, that sounds like a good idea. So she got together with another woman in town, and who became her partner. They went and charmed all the bankers in Hanover and got more money, and they purchased one of the last remaining houses in the business district of Hanover. It was just one block from the main crossroads in Hanover on Main Street and what was called Wheelock Street. And it was an old colonial house that was built in 1826. And it was right there in the middle of everything. And they renovated that house and made it into a restaurant. And it was charming. And so she named that Café La Fraise. She became the chef, because after she taught cooking for like 6 years, she perfected her repertoire and her techniques and her methods and so forth. And it was just a matter of scaling up to be able to, well, it wasn't trivial, I have to say, but it's a matter of scaling up and also organizing and planning for how many dinners of this you're going to need and all this other stuff. And she just basically took off and blossomed during the next 10 years in Hanover with her restaurant. And she became famous in Hanover. Everybody knew who Lynn Smith was. She was the young woman who was the chef at Café La Fraise, who made all that really good French food, and everybody knew her already anyway from the Parisian kitchen cooking school. And the kids liked it because because it was so central in Hanover, the kids would stop there on their way home from school, and all their buddies would come too. And after a while, the buddies, it wasn't Café La Fraise. Fraise means strawberry. And there was the sign, there's a big strawberry. All my son's buddies nicknamed it or dubbed the name Café La Frenzy Free, because they'd stop there for a snack on their way home from school. So Lynn could keep track of the kids that way a little bit, as much as you could teenage boys at that particular point in time. It was really good for me and I knew that was going to be the case. And I really encouraged this because I was so busy and the kids were growing up, and so we didn't have time for our relationship anymore. The honeymoon was over. And so I knew that she needed to be as busy as I was busy in order for this marriage to really survive and thrive. And that's exactly what happened. And the thing is that I'd stop there on the way to work. The restaurant was halfway between our house on Pleasant Street and the medical school where my lab was. And so I would stop there on the way in the morning and make myself an espresso. And I'd get a croissant fresh from the oven, and then we'd shoot the breeze about what was going to happen that day and whatever. And then I'd go on to work. And then I'd also stop there on the way home in the evening, just before the service started, and just to say hi, hello. All right, glad to see you're still here and so forth. And then I'd go home and be there with the kids. And then I'd make dinner, or we'd make dinner. It wasn't gourmet from Chef Kendall at all, but it was like macaroni and cheese, and hot dogs. Then Lynn would call me around 9 o'clock after the last dinner went out and ask me what I wanted for dinner that night. And I could order anything off the menu, or I could have the special of the day and so forth. And so I'd go in, and then we would, go talk over our mutual management problems that's when we had our relationship. So that was good. And that went on then for like 10 years, essentially. In terms of hormones and the immune system, the other good thing that happened when I'd first come back from France in the 70s, there was a fellow upstairs at Dartmouth Medical School, on the 4th floor. My lab was on the 3rd floor. His name was Alan Monk. And Alan Monk was an endocrinologist. And he'd been one of the premier, he still was, endocrinologists, because he had discovered the glucocorticoid hormone receptor. Glucocorticoids are steroid hormones made by the adrenal gland that sits on top of the kidney. And glucocorticoids regulate all over the body the metabolism of foodstuffs, carbohydrates, proteins, and fats. And they regulate the usage of all these foodstuffs and how the cells metabolize them and burn them and all this other stuff. And so consequently, there had to have been ways that the glucocorticoid hormones influenced all the tissues in the body, which it did: the brain, the muscles, the fat cells, everything. And so Alan Monk, who was an interesting guy because he was originally educated got his PhD as an electrical engineer or chemical engineer, I can't remember which. Anyway, he was an engineer. But then he took a postdoc at MIT in a lab there was an endocrine lab interested in hormones. And while he was there, he learned all about hormones and hormone receptors and that sort of thing. And then during the decade of the 60s, he came to Dartmouth and he started working on glucocorticoid hormones, and he devised an assay for the receptor for glucocorticoid hormones. Now, and he published that in 1968. So then I arrived at Dartmouth in 1973 for a year, and then went off to France, or 72, and went off to France and came back from France in 74. And that's when I met Alan. And Alan, because of the fact that lymphocytes were very influenced by glucocorticoid hormones, and I'd known that from my clinical training, because they today, and even then and now, they're used as drugs to treat inflammatory reactions, autoimmune reactions, and they also are lethal for lymphoid leukemia and lymphoma cells. So they profoundly influence, and I knew that because I had used them therapeutically when I was at the BCRC and so forth to treat the patients with leukemia and lymphoma. And Alan, when he set up his assay for glucocorticoid hormone receptors, the organ that he used to be the target organ was the thymus, which is, as you know, is the thing in the chest, collection in the chest where all the T cells go to get educated. And so they're all T cells. And if you take a mouse or a rat and you give them a big dose of glucocorticoid hormones, within 24 hours, the thymus just goes away. Everything dies and it just zoom. And so it was a good target organ for him to do these models, experiments and so forth. So when I met him, then we started, I said, well, that's interesting. And I learned about, we were just chatting about hormones and receptors and all that sort of thing. And it was in the late 70s, before we'd gotten into, or as we were getting into TCGF and IL-2 and all that sort of thing. And one of the very first experiments that we did together that Alan suggested was to see whether or not our human lymphocytes had glucocorticoid receptors. And so he taught us the assay, the glucocorticoid receptor assay, and all the intricacies, the ins and outs, and how to do it correctly. And basically, it's simple, but it's like anything else. You got to have a lot of technique, and so you have to do it right. And the thing is what you do is you have to have a radio-labeled hormone, so that you can trace it in different compartments and see where it is. So you incubate your radio-labeled hormone with your cells for a while until the hormone binds to the receptors, and then it comes to a steady state, and then you simply centrifuge the cells out, take off the supernatant, count the radioactivity in the supernatant, count the radioactivity in the cell button, and then you can calculate how much hormone is in the environment and how much stuck to the cells, and that's what's called the radio-labeled receptor binding assay, or radio-labeled hormone binding assay. So that's what he had reported in 1968, and that's what we did with lymphocytes, human lymphocytes. He'd only done it with rat thymus cells, and so we did exactly the same thing. And I didn't think this was such a great shake, so I expected the cells to have receptors. And then we decided, well, let's activate them with mitogens like phytohemagglutinin and PHA, and see what happens. And lo and behold, the number of receptors tripled inside the cells. And so that became my first Nature paper. We wrote that up. It was published in Nature in 1977. So I had 2 papers in Nature in 1977. That was sort of neat for a young obstreperous young guy like me. And then we went on. Now, subsequently, meanwhile, we were working on TCGF and LAF and all this other stuff. And I said, well, if TCGF is really the reason that the cells are growing, and we knew already because Peter Knoll back in 1960, one of the very first experiments he did once he realized that he could make the cells proliferate with PHA, is he threw in glucocorticoids to see if they would dampen it down, and they did. And he published that in 1961. So between 1961 and the late 70s, that's almost 20 years, and so everybody knew that they stopped this proliferation that was going on. They didn't know how it did it. Well, once we realized that this growth factor was the key ingredient, we could make the hypothesis that, well, either the glucocorticoids block the production of the growth factor, or they block the reception of the growth factor, its activity, or both. And so those kinds of experiments, once we had the TCGF assay, and we could measure the proliferation, those experiments were duck soup for us, and so we did them. And the answer was clear-cut, number one, and very simple. It blocked the production, but it didn't block the activity of the growth factor. Because if you put the steroids in, the glucocorticoids, and then added, it didn't, since it blocked production, you could give them the TCGF, and they'd still proliferate. So the experiments were, they were incontrovertible. So we sent those off to the JI, and they were published in 1979, and they were the forerunner in the early 80s. There were several different drugs that were created by other people. One was called cyclosporin A. Well, I should back up and say, so in addition to treatment of autoinflammatory diseases and leukemia and lymphoma, glucocorticoids were also used in transplants to dampen down the transplant rejection for kidneys, primarily at that stage of the game. But they needed other drugs. And so cyclosporin A came about within a year or two of our publication on glucocorticoids blocking IL-2 production. Cyclosporin A was shown to block TCGF production, and then another drug from Japan called FK506, and then another drug, and this happened in the first 2 or 3 years of the 1980s, called rapamycin. Didn't block the production of TCGF, but blocked the activity. And so what happened was that you could combine all of these drugs because they did similar things by dampening down the immune response, but they did it by different mechanisms of biochemical mechanisms of action inside the cell. So that means you could reduce the doses of the concentrations of these drugs so that you could get away with not very much toxicity over the body, but they would have tremendous dampening effect on the immune system. And that blew the lid off transplantation. That put the surgeons in the operating room. That created the whole subspecialty in surgery of transplantation. Because before that, the only thing that they could really transplant and have a reasonable amount of success were kidneys. But after this round of drugs in the early 80s came into being and became into use, and happened very rapidly. I mean, it was just like somebody turned on the faucet, and they're transplanting hearts, they were transplanting lungs, pancreases, all kinds of things. And so that was basically transplantation surgery and transplantation immunology. And it's really not a whole lot different nowadays. Using the same drugs that came about in the 1980s. Other ones have come in and so forth and so on to, tweak it a little bit and make it a little, make it work a little bit better and less toxic and so forth. But that was good. And to me, that was the first, I sort of said to myself, well, yeah, that's how the immune system works. But you see, the reason I keep harping on this whole hormonal thing is because most of immunology, they didn't really look upon the mechanistic aspects, the biochemical mechanistic aspects of all these things in the same way that I did. So I still had an uphill trek. I won't call it a battle. It was a trek to try to convince the rest of the immunological community that there was just a wonderful array of different kinds of applications, therapeutic applications, to the use of the knowledge that you can either block the production of the hormone, or you can enhance its production, or you can block its activity, or you can enhance its activity. And those are 4 different ways to modulate the immune response in your favor. And it depends on what you want to do. Now then, when it became towards the end of the 70s, and after we'd done the glucocorticoid experiments, the receptor experiments with Alan Monk, and then the functional experiments on TCGF production and so forth, and published them in 79, then from 1980 on, it was obvious what we should do experimentally, and that was, we had to find a receptor, because there had to be a receptor. How else was this crazy molecule going to do its deal? The other thing we had to do was we had to purify this molecule. We just couldn't continue to use this crude supernatant that we originally called Frankie's factor. We needed to have that stuff in a bottle. And so that was it. And that's what we embarked on. And while I was in France in the mid-70s, or early to mid-70s, Torg and I had discussed, how this erythropoietin molecule worked to cause more and more red cells to proliferate and red cells to be produced. And so, we batted the questions around and so forth, and it seemed to both of us there was only one way that it could happen, and that is that it had to happen via a receptor. And, I'm going to have to stop here for a second because the sun is coming in and it's really bright. Okay, so after we discussed this idea that maybe there was an erythropoietin receptor, I went to the library at the Institute in France. They had a good library, actually, and then nobody else was there. And so I spent a lot of time in the library. I read all I could get my hands on in terms of receptorology at that time. And like in the mid-'70s, it was hormone receptors were in its infancy. There were no molecular data as to what the receptor molecules were. The only thing that the endocrinologist had was the radiolabeled hormone binding to the cell. And there were two different kinds of receptors had been described at that point in time. Receptors that were on the inside of the cell, and those were all steroid hormone receptors. And the steroids, glucocorticoids or testosterone or estrogen or progesterone, they are all small molecules that are synthesized from a base of cholesterol, which is a multi-ringed organic structure of a lot of carbons and so forth. And the thing about these steroid hormones is that they can sort of slither in between fat and water. And they're called amphipathic because of that. So they can zip right through the cell membrane and go into the cytoplasm of the cell, and that's where the steroid hormone receptors are. And that's when Alan Monk did his glucocorticoid receptor assay. He showed that that's where it was on the inside, wasn't on the outside. However, there are other kinds of hormones, like insulin, for example, had been described at that stage, and two other molecules, epidermal growth factor and nerve growth factor. And they were like interleukin-2, TCGF, they were small proteins. They were too big and they couldn't scoot through the membrane and get inside the cell. So they had to have receptors on the outside of the cell, on the cell membrane. And that's basically what had been described at that point in time. And I learned about that. And so I figured that, erythropoietin was a small protein. We didn't know that, but we figured it was. And so therefore it had to have a cell surface receptor. And we did a series of experiments back in France in the mid-'70s that essentially showed that. And we put that in our first paper about the EPO assay. So when it came time to look for a TCGF or an interleukin-2 receptor, I was already well schooled in glucocorticoid receptors from Alan Monk. Plus, I was also, intellectually receptive to the idea that this is what it had to be. And so together with Alan, and then a new postdoc came into the lab who was a very good biochemist, trained at a well-known laboratory at Harvard in membrane and protein biochemistry. So he was very successful in doing separate procedures to purify IL-2, TCGF. And so we needed it radiolabeled. And so we would produce IL-2 in our cell culture systems in the presence of radiolabeled amino acids, the precursors of proteins. They would just become incorporated into the TCGF. And then Rich Rao was this fellow's name, and he would purify it. And then we could show that we had only pure TCGF radiolabeled with different biochemical techniques and so on. And then Alan helped us devise an assay for using our cell lines, our CTLLs, and then activated and resting T cells. And we showed that the resting T cells had barely detectable receptors, if any, whereas if you activated them with PHA or with a specific antigen, then they would blossom with receptors on their cell surface. And so we did a series of experiments that were, again, like falling off a log. I mean, everything worked. There were no scratch in the head business here. Everything we tried, everything we put together worked like a charm. And with a very short period of time, within about a year, we had data that indicated that the TCGF receptor, we still called it TCGF, we hadn't gotten transferred into the interleukin nomenclature yet. We could show that the characteristics of these receptors were all of the classic characteristics of a true hormone receptor as defined by the endocrinologists. And those characteristics were four. First of all, there were not an unlimited, there was a finite number of receptors on each cell. And that meant that if you then increased the concentration of the radiolabeled hormone, as you would then bind more and more and more, and then you would ultimately plateau because you could saturate them. No matter how much more you added, you couldn't find any more binding sites. So that was number one. Number two, the receptors the binding was stereospecific for the interleukin-2 molecule. If we used other things like EPO or other factors like LAF or whatever we could get our hands on and tried to compete the radiolabeled IL-2 binding, nothing would compete. It was very specific for IL-2. Thirdly, the concentrations that bound to the cells were in the range. They were identical with the concentrations that we could show in our assays that caused the cells to grow. So we could make the statement that they were physiologic hormone receptors. Now I'm sort of blocking on the fourth characteristic right now, but I'll think of it in a little bit and I'll come back to it. Anyway, so we had these things, and once we had all the characteristics of the receptor together, I ran up to Alan's lab and I said, Alan, so what we're dealing with is a true bona fide hormone receptor, right? And he sat there, he was 15 years older than I was. So he was the hair professor and I was just a young assistant professor kind of a guy. And he said, well, now you really can't call it a true hormone until you can show that hormone receptor system is under negative feedback and regulation. And that's true. I mean, for example, the glucocorticoid hormone is produced by the adrenal gland, but it's produced there because it gets a signal from the pituitary gland that sends a different hormone down to the adrenal gland to say, make glucocorticoid hormone. So the adrenal gland makes it, and when it gets to be a high enough concentration, that thing then turns off the pituitary gland up in the brain. So it's under negative feedback control. And this was an established precept and concept in endocrinology. And so I thought, he's absolutely right. So we designed a couple, some experiments as much as we could do. We only had two assays. We had the binding assay and we had the proliferation assay. But it was clear after we did as much as we could do, and we really couldn't convince ourselves that we could show that there was negative feedback regulation. And so, we did what we could do and we stopped doing that. But I still was out there on the stump talking about the hormones of the immune system. Most of the immunologists they were like dogs watching TV. They didn't know when to bark or not. They didn't know what that meant. They didn't have any education. They were ignorant. That's what they were. So it was up to me to educate them, as I figured. And so we published that in 1981, and actually October of 1981 in the Journal of Experimental Medicine. And that was another one of what is now one of the classics of immunology, because with Alan's guidance and teaching, and he basically became one of the scientific mentors that I had always been looking for, but I'd never found. And he was great. He was just wonderful. That paper became the prototypic paper for the next decade. And anybody that came up with a new interleukin and wanted to look for interleukin receptors for that interleukin, they just basically copied what we'd done in our paper. And right about that time, I had gone on a little mini sabbatical in the fall of 1981 to Lausanne, Switzerland, to Basel, Switzerland, I'm sorry, to work with a new friend that I had made in my travels and so forth, Max Schreier, who was this delightful German guy. And I went to work for him for 3 months and gave seminars all over Switzerland. And that was fun. I got, I would only sign on the train that had a knife and a fork, little symbol, icon in the thing, because they had a bar car. Switzerland's small. And so you'd have enough time for a couple of beers on your way to your next gig. And that was great. I had a wonderful time, and I met more and more people and so forth. And while I was there in Basel, and the reason I went there to work with Max is that Max was a B-cell guy, kind of a guy. And we both figured that, well, if there's a T-cell growth factor, there's got to be a B-cell growth factor, right? Because they proliferate too after they see their antigen and so forth and so on. So I went there, and he had all these T helper cell clones. He was the first guy so we cloned cytotoxic T cells. He cloned helper T cells for the first time. So he had all these helper T cell clones. And so I went there to see if I could identify a B cell growth factor that the T helper cells were making, because the reason they're helpers is because they help the B cells make antibody. And I always told Max, I said, well, you see, the B cells are stupid. They need help, and they do need direction from the T cells. Otherwise, they don't know what to do, Anyway, I was working on that in Max's lab. And during that time, I was looking for another postdoc. And I put an ad in Science and so forth, in Nature. And I got an interesting application from a young woman in England, Doreen Cantrell. And Doreen had gotten her PhD, just gotten her PhD, or was in the process of getting her PhD in Nottingham. That's where Robin Hood came from, and so forth. And so I wrote back. Her CV was interesting because after she finished her high school and had her baccalaureate, she took a year off and became an au pair in Thailand, in Bangkok. And I sort of thought, well, that's interesting. I mean, that's sort of a gutsy thing to do, If you're only like 18 years old or whatever, 17, and going off to Thailand. Anyway, I wrote her back because of that, actually, and so forth. And another thing was she was an immunologist and so forth. And I said, well, can you come over to Basel, and we can meet, and we can interview and so forth. So she said, okay. And so she came over to Basel, and we spent a whole day in the Institute cafeteria, essentially, drinking coffee and talking science, nonstop, from morning until turned into afternoon, which turned into then evening. And so then we went to dinner with Max and so forth. And when I was talking to her, I liked her right away. She she had this British accent. The Brits always sound smarter than the Americans just by the way they talk, So I liked that right away. And then the other thing was that she was very bright, and she was very quick, and she knew her immunology, history, and she knew the science really well. So I figured that, immunologically speaking, she had her head screwed on straight and knew what the questions were and knew what she wanted to do. And so then as time went on, I started asking her questions about, how movable she was. Could she, emigrate to the United States and so forth? And she said, no, that's not a problem. And really, she said, the only problem is my cat. I'm going to have to leave my cat behind. And I really like my cat. Well, that almost nailed it for me because of the fact that I'm a cat person. There's two different kinds of people. There are cat people or there are dog people. And I'm a cat person. And so was she. And so that was good. And so just as we were leaving to go to dinner, she said, she let it out that there was the other problem. There was no problem, but she was going to have to leave her husband behind too. And I had no idea she was married. She didn't tell me that. I didn't know that. But that clinched it for me. I figured that she'd really she's right on. She could leave her husband behind. And so she did. And she came to the States, and she came right at the time. And she had done her PhD work using the flow cytometer. Now, the flow cytometer was just recently introduced to immunology by a fellow at Stanford. And Len Herzenberg. And this revolutionized the technology of immunology, because prior to the flow cytometer, the only thing we could do as scientists would be to look down the microscope at a cell, at a population of cells. So there'd be millions of cells down at the end of the microscope. So, and we could sort of see what they looked like. And we could make slides of them then, and stain them with different histochemical stains and so forth. But that's about all we could do. The flow cytometer just basically took us up, several orders of magnitude up, because what the flow cytometer allowed one to do is to tag the cells with a fluorescent antibody that would bind to a molecule on the surface of the cell. So for the very first time in immunology, we could identify individual cells, and we could count the number of receptors that they had on the cell surface with this machine, together with a computer and so forth and so on. So we had single cell analysis and we had molecular analysis, and that was a revolution. And it was a revolution because of the fact that, I think, as I said before in one of the other videos, molecules are about 1,000 times smaller than a cell. And so that took us to the molecular level for the first time in immunology. And so also at the same time, just before Doreen got there, we had done a series of experiments collaborating with a group down at the NCI, Tom Waldman's group, because they thought they had an antibody that identified the IL-2, the TCGF receptor. So when they saw our paper out in 1981 in the fall, they immediately wrote us a letter and said, we think we have an antibody and so they asked me for some radiolabeled IL-2, TCGF. So I thought about that for a second, and I said, no, they'll just, to myself, I said this, the bubble, the cartoon, they'll just screw up the assay and we won't know anything. And so I wrote back and I said, no, send me the antibody and I'll do the experiment. Actually, I went down there and we sort of negotiated this question for a while, who's going to do the experiment. And I tried to explain to them that it wasn't like just trivial, and it wasn't just like falling off a log, and not any old immunologist could do this kind of an experiment that I could do, and so forth. So anyway, Tom at one point, we were discussing this, and Tom at one point left the room and came back with a styrofoam box. He says, "Here's the antibody." Gave me his box of antibody. So I went back to Dartmouth. I did the very first experiment, was clear cut. And that antibody blocked radiolabeled IL-2 binding, and we published another paper in 1982 in Nature. So that was just when Doreen arrived on the scene. And so now we had an antibody. We had she had the flow cytometry technology. And so we set about trying to see whether or not we could use this antibody to sort of map the distribution of receptors, TCGF, IL-2 receptors on the surface of T cells. And so over the course of a couple of years then, Doreen did a series of experiments We published several different papers. And it culminated really in one of the very, very best papers, if not the best paper that we ever published from my laboratory, which appeared in Science in 1984. And it essentially, it answered questions in terms of why there's such variability in the way cells grow in cultures. Some cells are very fast. It's like cells grow in culture like runners running the New York Marathon. So they all line up on the Verrazano Narrows Bridge in New York. The gun goes off and they all start to run. But there's guys at the beginning and there's guys at the end and there's a lot of them in the middle and so forth. And the fastest runners take off in the front, and then most of the crowd comes, and then there's these trailers that come back and finally cross the finish line five hours later, whereas the fastest do the 26 miles in only 2 hours. The cells grow exactly the same way. There's fast-growing cells and there's slow-growing cells. And the question was, why was that? And did it have anything to do with the IL-2 receptor? So the very first profile of the experiments from the flow cytometer that Doreen got, that showed a log normal distribution on the population of T cells. There were some T cells that had huge numbers of receptors, and then other T cells that had just a piddly number. And so we said to ourselves, well, I wonder whether there's any difference between the ones that have a lot of receptors versus the ones that don't have any or very few receptors. And the French would say bien sûr, which means of course. There was a difference. And the difference was that the guys with the highest density of receptors were the fastest. And when you would set them off by, say, adding IL-2 to the cultures, there were some cells, and then the highest receptor density cells, and they would just squirt through as fast as ever, and then there would be these laggards, and so forth. And so, in analyzing all the data, and people had known about this in all cells growing from bacteria to yeast to protozoan to all the nucleated mammalian cells and avian cells, they all were like this. But they couldn't go any further because they didn't understand the molecules that were responsible for causing the cells to grow, whereas we did in T cells and IL-2 for the first time in the history of science and so forth. And I didn't really realize that until I went back and started writing the paper. And then I read back to 1932, bacteria. The first paper was on bacteria in 1932, pointing this sort of phenomenon out. And over the course of all this 40 years, there are all kinds of theories to explain the variability of some fast cells and some slow cells and so forth and so on. And we basically put it out there. And it was remarkable. And then we said to ourselves, well, how does that work? So okay, you got more receptors. What does that mean? Well, what it meant is once the cells were occupied on the outside of the cell, biochemical signals were being transmitted through the membrane into the inside of the cell. And somehow, it took several hours. It didn't happen, it wasn't instantaneous. It wasn't like a nerve pulse causing the muscle to contract suddenly like that. It would take several hours for even the highest receptor density cells to start to replicate DNA. And so the duration of the occupancy of the receptor by the TCGF, the IL-2, was a critical variable. And if you knew that, and if you knew the concentration of the ligand, which was IL-2, and you knew the density of the receptors, you could predict which cells would be the fastest, which ones would be the middle guys, and which ones would be the slow guys. And that meant, in the end, that somehow cells could count the number of receptor hits that it got. And when it got to be the critical number whatever that was, let's say it's 1,000 or 10,000. It took, if you started with a cell that had low density receptors and you gave them a saturating concentration of the ligand, they'd still replicate their DNA and divide, but it would take them twice as long as the cells with the high density receptors. And so they had to be able to count the number of hits. And that's still, that phenomenon is still, we still don't understand how they count after, so what has this been? So it's been 40 years, 1980 or 1980 to 2020. And so I think we're gonna have to need some more young people to come along and solve this particular question. At the time that we were doing all these experiments, the glucocorticoid ones, the IL-2 receptor, and the cell growth, we published that as the IL-2 T cell system, a new growth model, a new cell growth model for people, because a lot of the people just interested in cell growth, nothing to do with immunology, they were using funny, they were using these mouse embryo fibroblasts that they thought were normal and all this other stuff, and so this was really revolutionary again. Well, about this time, another person came into my life that profoundly influenced me, and this fellow's name was Maurice Landy. And I knew who he was. I mean, he was basically the grandfather of our field of lymphokines. He had started out at the NIH, at the Institute of Allergy and Infectious Diseases in the 40s and 50s. And so I met him in the early 80s. And I had given a talk at a meeting, and afterwards, during the break or whatever, he came up to me and introduced himself to me and so forth. And the first thing he said, Kendall, I can help you. I said, really? How can you do that? And he said, I can help you write your papers. At the time, he was retired from the lab, but he was an immunologist, so he knew the business. And he was working for Academic Press, who was one of the big academic and particularly science publishers, as an editor. And so he just that's what he did. He read papers and he, edited them. And he said, if you send me your next paper when you think you've gotten it just about the fact that it's, you're happy with it, send it to me. So I did. I can't remember which paper that was, but it was back in the late 70s. And when we were publishing all of our best work. And I got that paper back in return post with red marks all over it. I felt like I was back in English class, And the thing is that he didn't change what I said, what the gist of the paper was and so forth, what I was reporting, but he changed how I said it. And, he would take the first part of the sentence and put it over into the back, or vice versa. He would use different adjectives and adverbs. He would clean up the poor grammar with dangling modifiers all over the place and so forth. And he really made the papers a piece of literature. It was wonderful. And I sent him every paper and every grant and everything I wrote for the next 5 years. And that's when I learned how to write. I didn't learn how to write in school. I was going to be a doctor, and I didn't think I was going to write anything except prescriptions, and I could just scribble them, And so, I used to write my English compositions when I was in college the night before. I'd just sit down and I'd spew off whatever, and I'd hand it in. So I'd have something to hand in the next day, and I always got C's on it because I'm C-, sometimes C+. Never got a B. But Maurice Landy made it fun, and I understood for the first time how creative this process was. And now that I'm trying to become I'm a wannabe artist, it's exactly the same process, because let's say you're going to draw a horse, whatever. And so you make a drawing, and it looks pretty good. Looks like a horse. Then you put it down, and the next day you come look at it, and you say, well, the neck's too long, or whatever, and so you modify it. You erase it, and you modify it, and so you work on that thing until you get it the way you want it. But you do the same thing in writing. So you write a paragraph, with what do they call it? The first sentence is the whatever, and then the whole thing thereafter, and then you can write another one in another paragraph, and so ultimately you get done writing, and then you set it aside because you've got other things to do and so forth, and so then, but you think about it. You think about it all day long and all night long. I mean, and what happens to me is it all sort of gets reordered while I'm sleeping. I wake up in the morning and I look at that paragraph that I wrote, and I rearrange it. Rearrange the sentences, I rearrange the words, I rearrange the paragraph to get it to shape it, to get it, make it look like a horse. And it's really a fun process, and so that's what I do now. Now, whatever. So Maurice Landy was great, and he was a tremendous influence on me. And he kept saying to me over and over and over again, he said, Kendall, a scientific report has got to be concise, tightly integrated, so that the beginning, the intro, and the methods, and the results, and the discussion, all those sections are tightly integrated. So when you're reading through, that are really together. And he said, it's got to be persuasive. By the time the person reads through your whole paper, they have to be utterly convinced that this is the best thing since crushed ice. And so I try to do that with every paper, everything that I write, essentially, because it keeps ringing in my head. Now there's one other person that came into my ken, as the Israelis say, during this time period, the same, early 80s, late 70s and early 80s, and that was a fellow by the name of Ellis Reinhertz, who we became friends, and we're still friends today, and we discuss the state of the world over the phone a couple of 3 times a week. He calls me on his way to work in the morning. He's still working. And he calls me on the way to work, and from his Mercedes-Benz on his car phone, and complains about his lot in life as a working scientist. And so I try to give him advice. Anyway, it was the end of the 70s when I first met him, and we were placed together on a symposium, and I happened to talk first. I talked first, and then I sat down in the audience. And I listened to Ellis, and he had just done a series of experiments with newly available monoclonal antibodies that would react with T cells, and so he was using these antibodies to characterize which T cells and these antibodies would react with. So they would bind to the surface of the cell, and he used the flow cytometer, among other things, to show that they reacted with the T cells. And he reported in the late 70s 3 different antibodies that ultimately became called, they initially were called T3, they were just numbered, T4 and T8. It turns out the T3 antibodies reacted with all T cells. The T4 antibodies reacted with about 1/3 of them from peripheral blood of humans. And the T8 antibodies react with 2/3 of them, and they were mutually exclusive. So this is what he talked about in this symposium. And I'm sitting there listening to this guy, and it was the first time I'd heard this story, or at least in the human. Similar things had been done a couple of years earlier in the mouse T cells. And so I listened to him, and I said, this guy's really smart. He's almost as smart as me. I'm not going to compete with him. I'm going to make him my friend. And that's what I did. And we went out to dinner that night, became fast friends for the rest of the time. It turns out, soon after, there was a meeting, the International Immunology Congress happened in 1983, in Kyoto, Japan. And Alice and I traveled there together, and we roomed together, and we talked science all the time and so forth, and went out late at night drinking and stuff like that. And he had just described in the spring of 1983 the first experiments that identified the T-cell antigen receptor. His lab and the other people didn't quite understand what the T-cell receptor did versus what the IL-2 receptor did. And so I explained it to him. He told me he was educable. I thought that was good. I'd try him out on this. And so I told him, I said, listen, this is the way it works, Ellis. The T-cell antigen receptor is the first thing that happens, and it causes the cells to make TCGF, IL-2, and to express receptors for it. And he said, okay. And I said, and he had all these reagents that he had created, because he made some of the first human T-cell clones that reacted only with one particular antigen, and that's why they were clones. So I proposed some experiments. So we got together afterwards. Doreen was with me, and she got together with Stefan Moyer, who was one of Alice's postdocs, and Becky Husey, who is the chief tech in Alice's lab, who was really gifted in terms of culturing these T cells and so forth. And we did a series of experiments that essentially showed what I just said, and that is that what the T cell receptor does versus what the IL-2 receptor does. So we published that in the Proceedings of the National Academy of Sciences. It came out in early 1984. And basically what that paper says, that it's IL-2 dummy that makes the cells grow. That's the end of chapter 4. And I figured that by this time now, we really had nailed the story together, essentially, in terms of it being a hormonal system that is regulating the immune response, the T-cell immune response. And as time went on in the 80s, more and more interleukins were discovered, interleukin 4, interleukin 6, that were very important for B cells, to make them proliferate on the one hand, and also to make them start to make antibodies. There were factors, interleukins just discovered that activated natural killer cells. So there's all, it might sound like it's alphabet soup, and it really is. It's alphabet soup. It's pretty complicated. It's not easy to sort all this business out. But that's the immune system at this stage of the game, and everybody's been working on it ever since. So I'll stop there. Thanks.